You know, our immune system is this incredible built-in defense force, but for centuries, it was pretty much a black box. Now, though, we've gone way beyond just watching it work. We've actually learned how to harness it, how to train it for battles it hasn't even fought yet, and how to use its own weapons as these high-tech detective tools. So let's dive into how we do it.
It really sounds like something straight out of a sci-fi movie, right? But this one question, this one idea, is what kicked off a massive revolution in medicine. It's the core concept behind one of the single greatest public health achievements in all of human history: vaccination.
So here's how we're going to break it down. We'll start with the basics of harnessing our immune system and see how vaccines work like a training program. Then we'll check out the modern vaccine toolkit. After that, we'll switch gears and look at how immunity can be used as a detective, focusing on those amazing diagnostic antibodies.
Right. So how do you take this unbelievably complex biological system and turn all that knowledge into life-saving technology? Well, it all starts with control, learning how to purposefully guide our immune response.
Let's kick things off with vaccines. Really, the best way to think about a vaccine is like a training program, or maybe like a military drill, for your immune cells. It gives them a totally safe way to practice fighting a very specific enemy. So if the real thing ever shows up, they are more than ready. Now, this story starts way before we even knew what a virus was. Back in the 18th century, people were already trying out different forms of inoculation. But the huge breakthrough came from an English doctor named Edward Jenner. He made this incredibly sharp observation. Milkmaids who got the pretty mild disease cowpox just didn't seem to get the deadly scourge of smallpox. And that led him to a truly revolutionary experiment.
So why on earth did this work? Well, by introducing the much milder cowpox virus, Jenner's technique essentially gave the immune system a practice run. It started producing antibodies and, this is the crucial part, it created long-term memory cells. So when the real smallpox virus came along, those memory cells were ready to go, launching this super-fast, powerful response that just wiped out the threat before it ever took hold.
And that process right there, that's the basis for all vaccines. And in a really cool tribute, the term vaccine itself was actually coined from vacca, which is the Latin word for cow. I mean, think about that. A simple observation about dairymaids led to a technology that would one day completely eradicate smallpox from the face of the earth. Amazing.
Jenner's method was an absolute game changer. But today's science has gone so far beyond just using similar viruses. We now have this whole toolkit of really sophisticated vaccine strategies, and each one is designed to train our immune system in a slightly different way. Two of the main strategies really highlight a fundamental trade-off. First, you've got attenuated vaccines, which use a live but seriously weakened microbe. These create a really strong, often lifelong immunity because they're so close to a real infection. Then you have inactivated vaccines, which use a completely killed version of the microbe. They're incredibly safe. They can't possibly cause the disease, but the immune response is a bit weaker, which often means you need booster shots to stay protected.
And you can see this diversity in action right here. The measles vaccine uses a weakened live virus. Polio uses a killed virus. And for something like hepatitis B, we don't even use the whole virus at all. We just use these tiny pieces of it, called antigenic fragments, that are just enough to get the immune system's memory going. It just goes to show you that every disease really requires its own tailored approach.
Okay, so we've seen how immunology helps us prevent disease. But believe it or not, that's only half the story. The other massive application is in detecting disease. We can actually take the immune system's own tools and use them as these little biological detectives to find clues of an infection or other health conditions.
The star players here are antibodies. Because they are so incredibly specific, they're designed to bind to just one particular target, or antigen, they are absolutely perfect for this job. Scientists can do things like attach a fluorescent dye to an antibody, and if that antibody finds its target in a patient's sample, it literally lights up under a microscope, giving us this crystal clear visual confirmation.
But the real game changer for diagnostics was the invention of monoclonal antibodies, or mAbs for short. Instead of just getting a random mix of antibodies from an animal, scientists figured out a way to produce a single perfect type of antibody, and they could do it endlessly. This gave us a virtually unlimited supply of ultra-specific biological detectives for all kinds of diagnostic tests.
So how do we actually use these maps in the real world? Well, it all comes down to their perfect lock-and-key specificity. And one of the most common and powerful methods is a test you might have heard of called ELISA.
The process is just so elegant. First,
Antibodies that are designed to catch a specific antigen are stuck to a plate. Then you add the patient's sample. If the antigen is in there, it gets caught. Everything else just gets washed away. Next, a second antibody, this one with an enzyme attached to it, is added. It binds to the antigen that's already trapped, creating a little sandwich. Finally, a chemical is added that the enzyme can convert into a color. If the color changes, the test is positive. Simple as that.
You can see that antibody sandwich perfectly in this diagram. The patient's antigen is trapped right between two super-specific antibodies. And that little enzyme on the second antibody, it's like a signal flare, just waiting to light up and tell us that we've found our target. It is an incredibly sensitive and specific system.
And you've almost certainly come across this technology without even realizing it. A home pregnancy test? Yep, that's an immunologic test using monoclonal antibodies to detect a hormone. That rapid strep test you get at the doctor's office? Same exact principle. It's also the technology we rely on to screen our blood supply for viruses like HIV, which is what makes transfusions so incredibly safe today.
So, when you just step back and look at the big picture, it's pretty incredible. By learning the language of our own immune system, we've developed these two monumental capabilities: the power to prevent devastating diseases before they even start, and the power to diagnose active problems with just astonishing speed and accuracy.
It really makes you wonder, doesn't it? We've learned to train our body's defenses and even use their own weapons as diagnostic tools. As our understanding of this ridiculously intricate system gets even deeper, what's the next frontier? What's the next breakthrough that's going to be unlocked by our own biology?